Bringing new diabetes drugs to market is challenging, takes years, costs a fortune and often fails. In early human phase studies, efficacy is measured based on repeated C-peptide sampling. But there is an inexpensive alternative. It requires no blood sampling, and with IRB approval, all the necessary components just might be in your waiting room now. We call it AIDclamp. The components are straightforward: an experimental protocol, an insulin-requiring volunteer using a DIY AID system, and here we show our standard, which is an Omnipod pump, a Dexcom sensor, and a smartphone running Android APS. Then software that collects insulin delivery such as Gluco, Tidepool, or NightScout, and finally software that provides timed calculation of insulin delivery. We love the NightScout interface, but even more, we love its reporting. And here in the red rectangle, we show 6 hours constituting the test meal used in our protocol. At 0900, a pre-bolus is given. At 0920, a 77-gram meal is taken. And then you can see not just the rise in sugar, but the blue line showing insulin on board and the red line showing carbs on board. In terms of our experimental protocol, the proof of concept is before you as ADA poster 980 for 2024 June, in which AIDclamp captured reduced insulin requirements with dual agonist therapy with repeated test meals. For the example I'm going to show in this slideshow, the investigational new drug causes beta cell proliferation. It's a hypothetical. The drug's given daily as an oral agent for 12 weeks, and then every 4 weeks for 20 weeks, serial mixed meal testing is performed. The hypothesis is that reduced insulin infusion over those 6 hours over 20 weeks reflects increased endogenous insulin capacity. Here we have the software interface that does the insulin calculation, and you can see in the second entry where this is where NightScout URL is placed, the start and end times for the test meal, the time zone, click submit, and the total insulin infused is 24.3 units of insulin. Here we have the graphical depiction of the hypothetical with 6 curves, the one on top at baseline, and each curve showing less insulin infused over the 6 hours, such that at 20 weeks, the total insulin infused is less than half the starting dose, and in our hypothetical, it would have been 24.3 units. So the decrement of 12 units is attributed to increased endogenous insulin infusion. So why AIDclamp? It's inexpensive, the protocols are simple, the timelines are short, it's convenient to participants with no blood draws, and there's no lab processing time.